In today's Crystal Formulation Classroom, we will cover factors affecting permeability. Chemical composition is one of the critical parameters of permeability. Many drugs exist in different ionization states depending on the pH of their environment. The ionization state can affect a drug's permeability through biological membranes. For example, charged molecules may have difficulty crossing lipid membranes, which are primarily permeable to nonpolar substances. Lipophilicity refers to the ability of a molecule to dissolve in fats, oils, and other nonpolar solvents. Lipophilic drugs tend to have higher permeability through the lipid membranes compared to the hydrophilic drugs. This property is important for drugs intended to reach intracellular targets. Some drugs may undergo chemical reactions or degradation processes when exposed to the physiological conditions. Degradation products or metabolites may have different permeability properties compared to the parent drug molecule, impacting their bioavailability and efficacy. As covered in our last episode, the solubility of a drug in various solvents or physiological fluids can impact its permeability. Drugs that are highly soluble in water may dissolve readily and pass through biological membranes more easily, while poorly soluble drugs may have limited permeability. Another factor is the size and shape of a drug molecule that can affect its ability to permeate through the biological barriers. Nanoparticles and microparticles with smaller size can penetrate biological membranes more efficiently compared to larger particles. This enhanced permeation across the epithelial and endothelial barriers can improve drug delivery to targeted tissues or organs, especially for drugs intended for systemic circulation or site-specific action. Smaller particles have a larger surface area per unit mass compared to the larger particles. This increased surface area can enhance dissolution rate of the drug, leading to faster release of the active pharmaceutical ingredient and potentially improving its bioavailability. Passive and active transport are two fundamental mechanisms by which pharmaceuticals permeate through biological barriers, such as the cell membrane. Efflux transporter, such as P-glycoprotein, is expressed in various tissues, including the epithelial cells of the gastrointestinal tract, the blood-brain barrier, the liver, kidneys, which actively pumps drugs out of the cells, limiting their intracellular accumulation and reducing their bioavailability and efficacy. These transporters play a significant role in regulating drug permeability across the biological membranes and can impact the drug efficacy and pharmacokinetics. Understanding interplay of the chemical composition, solubility, particle size, and efflux transporters affecting permeability is essential for drug development, formulation design, and optimization of the drug delivery systems to ensure adequate bioavailability and therapeutic efficacy. Thank you for joining us in this episode of Understanding Factors Affecting Permeability. Stay tuned for our next episode where we'll dive into the common methods to enhance solubility.